All right, continuing part 2. We're on the third key feature of the adaptive immunity, which was distinguishing self from non-self. And in the human body, you have to think of this. The human body contains thousands of different molecules. Each of them have a specific 3D structure that is capable of generating an immune response. So therefore, every cell in the body, there's a tremendous number of these antigens to help fight off these pathogens. Let's just hope that our immune system can recognize its own body's antigens and not attack them. So the body gives them basically a test. It's called clonal deletion. And this test is on B and T cells. And any immature B or T cell that shows the potential to mount a strong immune response against self-antigens will undergo programmed cell death, apoptosis, within a short time. If this test, this clonal deletion process, fails, then that will lead to an immune response or autoimmunity. There are 2 autoimmune diseases that we're going to cover. The first one is called SLE, which is known as lupus, systemic lupus erythematosus. And in these individuals, the antibodies can cause serious damage when they bind to normal tissue antigens. And within this damage, it forms large circulating antigen-antibody complexes. These huge complexes get stuck in tissues, and it provokes inflammation. The other autoimmune disease is Hashimoto's thyroiditis. It's most common in women, older women. The immune cells attack the thyroid, and because of that, it results in fatigue, depression, weight gain, and other symptoms. All right, the last key feature of the adaptive immunity is immunological memory. After responding to a particular type of pathogen once, it will remember that pathogen, and then it will respond faster to that same pathogen or threat in the future. So immunological memory saves us from repeat of these, bad infectious diseases that we might have had as a child or when we were younger. During the first exposure to the pathogen, that is called primary immune response, and then the second exposure would be called secondary immune response. In primary immune response, we have activated lymphocytes that will divide, and they'll differentiate to produce 2 different types of cells, daughter cells. One is called the effector cells, and these actually carry out the attack on the antigen. Effector B cells, which are called plasma cells, are going to secrete antibodies. Effector T cells are going to release cytokines. Effector cells only live a few days. Then you have memory cells that are produced. These are the cells that are long-lived, and they retain the memory. They hold and they remember. And what they do is they start dividing on short notice to produce more effector and more memory cells. And so memory B and T cells can survive for decades. In secondary immune response, once they already know, they've already fought off this pathogen, the second time around, it's more rapid and it's more powerful. The memory cells that bind with that antigen proliferate, which means they grow, and they launch a huge army of plasma cells and effector T cells. The principle behind being vaccinated is to trigger the primary immune response, and then that will prepare the body to mount a stronger, quicker secondary response if it needs to. In other words, if it encounters that actual pathogen again. Okay, so talking about the adaptive immune response, there are 3 basic phases for this immune response. Number 1 is recognition. The organism discriminates between self and non-self so that we can recognize these pathogens in our body. Phase 2 is activation. The recognition event will lead to mobilization of cells. These cells will move in, and these molecules will fight the invader. And then phase 3 is the effector phase, where the mobilized cells and the molecules will destroy the invader. So you have this picture here. You need to label the first phase as recognition phase in your box. And you can see you have free antigens. You have an antigen-presenting cell that will bind to a helper T cell. So a T cell that has the specific receptor will bind to the antigen on the presenting cell. The T cell then makes these cytokines which are going to stimulate other cells to divide. Then, in the activation phase, the specific B cell that binds the antigen is stimulated to form a clone. These B cells form specific antibodies that will bind to more free antigens. So the one on the left, when a B cell is involved where we are making antigens, that is called humoral immunity. You need to label that on your diagram. And then on the right, if a T cell is involved for the activation phase, the T cell that binds to the antigen on the antigen-presenting cell is stimulated to form a clone, so the same thing is occurring. And then in the effector phase, the T cells bind and destroy the cells that are bearing the antigen. If the T cell is involved, then it is called cellular immunity. Please label that on that side of your diagram. All right, so we're going to go into a little more detail on the humoral immunity. For the humoral response, the basis is B cells. There are billions of B cells, and they will survive the test of clonal deletion. And then the ones that survive are going to be released from bone marrow into the circulatory system. Remember, B cells have a receptor protein on its cell surface that is specific for a particular antigen. It gets activated by antigen binding to this receptor, and then after stimulation from a T cell, it produces a clone of plasma cells that make antibodies as well as memory cells. So your diagram is already labeled here. Here we have our antigen up here, and here is our B cell, a whole bunch of B cells. And they all have a specific antigen, and so obviously this antigen will bind to this particular B cell. It's specific. Each B cell makes a different specific antibody and displays it on its cell surface. So this B cell makes an antibody that binds this specific antigen, which is obviously different. The specific antigen also binds to a T cell, which stimulates the B cell to divide, resulting in a clone of cells. We're just getting a whole bunch of these cells. These cells eventually will make plasma cells and memory cells. Primary immune response, some cells will develop into plasma cells, which are the effector B cells, that will secrete the same antibody as the parent cell. Potentially, we have a secondary immune response. A few cells develop into non-secreting memory cells that will divide slower. All right, let's go into a little bit about antibodies. Antibodies are also called immunoglobulins. Two of the polypeptides are identical light chains, and two of them are identical heavy chains. They are held together by disulfide bonds, which we learned back in Chapter 3. Each chain has two different regions, a constant region and a variable region. The constant region is when the amino acid sequence determines the general structure and function of the immunoglobulin. The variable region is when the amino acid sequence is different for each specific immunoglobulin. So we have this diagram. It's all labeled for you. Let's go back to the next page first. There are two antigen binding sites making the antibody bivalent. This allows the antibodies to form large complexes within the antigen. Bivalent. So here we have our light chain. Also light chain over here. This is the variable region of the light chain. And this is the constant region of the light chain. Same thing with the heavy chain. And you can see all these little S's here. Those are how they are bonded together. Disulfide bond. Okay, for immunoglobulins there are 5 classes. Immunoglobulins are abbreviated as IG. These 5 classes of immunoglobulins differ in function and in the type of heavy chain. IgG is secreted by B cells and constitutes about 80% of circulating antibodies. IgD is an antibody where the cell surface receptor on the B cell. IgM is the initial surface and circulating antibody released by a B cell. IgA protects mucosa and epithelia, which is exposed to the environment. And IgE binds to mast cells and is involved with inflammation. And this will end part 2. Please move on to part 3 of the podcast.